Okay, good afternoon, all. So, as you know, that today's lecture is about the guidelines and recommendations for the review of literature. So, because we know, like when we are starting our PhD, when we are trying to figure out what should be the topic of my research, and that topic, that research you're going to carry ahead for next couple of years. So at that time, you might have lots of questions like how to start my research, what should be the research topic, then on what areas I should gather more information, what should be my uh, experiments, how do I interpret the experiments, what should be the subsections of my work, or whenever you are designing your thesis, then what should be the chapters and then subsections of each and every chapters. So there might be lots and lots of questions when you are starting your PhD. So in order to answer all these things, whenever you are starting your PhD, the most important aspect of your PhD is review of literature. The beginning, the very beginning of your PhD journey is review of literature. So today I'll be sharing you some guidelines and recommendations to do the best review of literature. I'm Dr. Saugata Ghosh, acting as Associate Professor, Department of Microbiology, RK University. I'm also serving as Associate Editor of Current Microbiology and Associate Editor of Frontiers in Chemistry. These are two international journals. At the same time, I'm also serving as reviewer of 127 international peer-reviewed journals. So whatever knowledge, whatever experience we have gained while doing this editorial work, I'll be sharing some of those insights with you also, because when one of the most important integral part of a PhD journey is publishing the paper. So when the papers come to us, when you submit a manuscript, we review the manuscript or we take the decisions, then why those manuscripts are rejected, why some manuscripts are accepted, why some manuscripts undergo revision. So all these small aspects also I'll try to cover up a little bit in this review of literature, because that is altogether a different lecture, how to prepare a manuscript. So that is also very interesting. But today, since we have time limitation, so our today's topic is based on only the review of literature. So now, when you look into the process of review of literature, you should think like why or what is review of literature all about. So first thing that we should know is what is meant by literature review. What is literature review? You should know this thing thoroughly. Second is why we should do literature review, the functions of literature review, why literature review is so important. And next is how we should do the literature review. So the steps of literature review. So all these three questions, that is what, why, and how. So all these three questions are very important when you start your process of literature review. Now, when you are looking at this review of literature, sometimes we call literature review or review of literature, it is the documentation of a comprehensive review of published or unpublished work. So when you club up all information, now these informations can be from which sources? Either they are published or they are yet to be published, not published yet. You can also take those. So when you club up all this information, when you gather all this information from different published and unpublished work, from secondary sources of data, in the areas of a specific interest to the researcher, then we call it as a review of literature. So here you have seen three keywords. What is the first keywords? First keyword is published work. So all those articles, those are published. Those are found in the public domain. You can select those as source of some data. There are some researches, there are some data which are not displayed openly in the public domain. So those are unpublished data. You can get it in some lab, you can get it in the thesis. You see so many dissertation theses are there, PhD thesis, master's thesis, lots of theses are there in the department. And all those theses, all those works, they are not published, but they are documented. They are documented in the form of a thesis. So you can refer to those work also. Now, when you are referring these are not your work, mind it. These are the work that are already reported by other researchers. So this becomes your secondary sources of data. What is the primary source of data? Primary source of data is your own work. When you work, you do some research, you generate some data, and you deduce some conclusion from your data. That is the primary source of data. But review of literature is you are reviewing the data, but which data? Not your data, the data that is either published by some other group or the data that is reported but yet not published, the data that has been generated by somebody else. So, whenever the data is not yours and you are reviewing it, you are collecting it, you are trying to do some research based on those data which are not published and not yours, then these data are called secondary sources of data. Next, what is the another aspect of review of literature? The literature review is an integral part of the entire research process. It is not that you are doing this review of literature just in the beginning, say first 1 month, 2 months, 3 months. It's not like that. It's the integral part of the entire research process. So suppose you are doing a research for 3 years, whole 3 years you have to do literature review. Why? Because now you are doing the literature review to design your project. After 6 months, you want to do an experiment. So how to start the experiment? What instrument you have to use? What chemicals you have to use? Again, you have to refer to somebody else's paper. After 2 years, you are doing some experiments. And then when you are doing some experiments after 2 years, then also in order to know about more about the experiment, what kind of process they are using, what temperature they are using, what pH they are using, again you have to refer to some paper. And at the end of the 3 years, when you are publishing your paper, then you have to again review with others paper because you have to compare your data, whether your data is superior to others report, or your data is inferior to others report, or your data is at par the others report. So throughout your journey, whether at the beginning of the journey or at the middle of the journey or at the end of the journey of your research, entire phase you have to do literature review. So this is a very important aspect of your PhD. And that's why it makes a valuable contribution to almost every operational step. Every operational step means in order when you start a research, first thing is to find out the research gap. That this work has been done and this work has not been done. So that means there is a scope, I can do some research on this area. So how do you know what work has been done and what work has not been done? By reading others' papers. So that means when you find out your research problem, research gap, you have to do research review of literature. When you are designing your experiments, you have to do the review of literature. When you are writing your manuscript, when you are reporting, when you are concluding your research, then also you have to do review of literature. So it is an integral part that contributes to almost every operational step of your PhD. Next, what are the functions? Why we need to do this review of literature? Why it is so important? First thing is it brings clarity and focus to your research problem. Now, suppose your guide tells you to do research review of literature. First month it goes like that, second month it goes like that, say third month it goes like that. So you might be thinking that your guide is neglecting you or your guide is just trying to avoid you or ignoring you. It's not like that. Whenever your supervisor is telling, no, it is not complete, you have to do some more review of literature, you work on this, you find out this. Every week you go with your review of literature and your guide, your supervisor tells, no, this is not complete, you do something like this, you search on this. So more you search, more you do the review of literature, the more clarity it gives on your research problem. Otherwise, if your review of literature is not complete, you have thought that you think that a particular research has not been done. Suppose you are from organic chemistry, biology, pharmacy, and other things, and you think like I will synthesize, I will derive, or I will extract a drug from tulsi, Ocimum sanctum, that will protect that will protect against pneumonia, so Klebsiella pneumoniae bacteria. Now you have started gathering the tulsi plant, Ocimum sanctum plant. You have started extracting the plant. You have started isolating the compound. You have started identifying the compound with the help of NMR, IR, and other things, GCMS. And finally, you use that compound to the biological activity against Klebsiella pneumoniae. You find out MIC value, minimum inhibitory concentration. You find out MBC value, minimum bactericidal concentration. Then you put it in mouse and check whether it can protect the mouse from pneumonia. So all these things you have already done, and finally you have got very good amount of data, and then you are going to publish. But the editor rejects it, rejects your publication, rejects your manuscript. Why? The editor tells that 10 years back already this work has been done. People have already reported Ocimum sanctum's effect on pneumonia, or Tulsi mediated cure of pneumonia. Some people have already reported this. So, what work you have done so far, that is all lost. You have done a very good quality of work, but it has become a mere repetition or redundant research, just a repetition of somebody else's work. So, why you have come to this problem? Because your review of literature was not complete. You have missed that paper, the paper which already reported Tulsi's effect on pneumonia. If you had already done this literature search properly, then you would have got this paper, and then you think that, oh, this has already been reported, Tulsi's effect on pneumonia, so I should think something different. Understood? So this brings clarity. If you do not do review of literature, then it will not give clarity what work has already been done, what work has to be done. So this clarity is very important, and focus to your research problem. So if this is done, then what new I can do? So you can focus on your research problem. So this can only be possible if you do a thorough literature search. Next is improve your methodology. So you want to find out Tulsi's effect on Klebsiella pneumoniae, on a bacteria. Now, are you going to use the whole Tulsi extract, or you are going to extract it after doing water extraction? You are going to extract, or what are the different other types of extraction? Can you give heat? If we can give heat, then what kind of extraction is possible? Then you find out, oh, Soxhlet extraction is very good for heating. If you want that I do not want to damage my compounds by heating, then what? Then supercritical fluid extraction is there. If you think water is not a good extraction process, so let me extract in other solvent, then you can prepare tincture, that is in ethanol. If you think moving or shaking is not a good methodology for extraction, I just want to keep it steady, then maceration is a good extraction. So there are different types of extraction process. How do you know all these things? Which method is best? Which method is good for Tulsi? Which method is good for antibacterial effect? The methodology will come to you. You will get more knowledge about methodology by doing review of literature. Otherwise, you will think, I'll extract. What is the method of extract? Just by putting the powder in the water and boiling, that is the only method of extraction. But no, there are hundreds of methods of extraction. Different people are extracting in different solvents. Different people are extracting in vacuum. Some people are extracting in heat. Some people are extracting in cold. Some people are extracting with carbon dioxide, liquid carbon dioxide. So there are so many different methodologies for extraction only. Understood? Once you extract, then you have to purify the compound. the best compound. How do you do the purification? There can be TLC, thin layer chromatography, there can be column chromatography, there can be HPLC, high performance liquid chromatography, there can be analytical HPLC, there can be preparative HPLC. Okay, so there are numerous methods for separation. Then there can be solvent fractionation. By separating funnel also you can just do some fractionation. So there are numerous methods for extraction. Once it is extracted, then you have to find out whether it is pure or not, and then you have to find out its structure. How do you find out the structure? NMR, IR, GCMS. Now there is GC-TOF-MS. Okay, so there are numerous UV. So there are numerous different methods that you are going to use. How do you know these methods? Which method is best for extraction? Which method is best for isolation? Which method is best for identification? By doing the review of literature. So it improves your methodology. Next is broaden your knowledge base. What is broaden your knowledge base? That means when you are searching, then you see you cannot get one paper on Tulsi extraction. You will get hundreds of paper. Somebody would have extracted Tulsi from the leaves. Somebody would have extracted Tulsi from the root. Somebody would have extracted Tulsi from the stem. People have used different parts. People might be using different solvents, water, methanol, 70% ethanol, ethyl acetate, chloroform, N-hexane. People might have isolated with different types of columns. Okay, silica gel, different types of TLC, different types of HPTLC. So different other methods people have used. So how will you get all this knowledge? From review of literature. And then contextualize your finding. What is contextualizing your finding? Contextualizing means how you can correlate what you want to find out and what are you actually finding out, whether it is superior or inferior with the previous findings. So you have to contextualize. Okay, what is your objective and what are your methods? So if the objectives are matching your methods, then that is contextualization, and then you get the results. So these four things are very important for why you want to do the review of literature. First one is clarity, second is methodology, third is knowledge base, and fourth is contextualization. Okay. Next, let us come to the clarity and focus to your research problem. So when we are finding out the clarity, as we know that a review of literature brings clarity. It makes the idea more clear. It helps you to focus on your research problem. So you cannot effectively start the literature review search without an idea of the problem. First thing, when you would start the review of literature, first thing that should be with you is your problem. So this problem, either you can decide yourself, but it is difficult. So that's why a problem is generally decided by mutual discussion between a fellow or a PhD scholar and the supervisor. You have to sit with your supervisor, you have to take his appointment or her appointment, and then you discuss that what should we do, what can be done as the part of the PhD, what should be the title of the PhD, what should be the problem. So once you have this idea, what is your research problem, then you can start your review of literature. Now, review of literature at the same time can help you to shape your research problem. How? Because when you do some review of literature, then you know already these things are already done. Now, you should not repeat this again. Understood? There is no need to repeat this again. You have to think, you have to design, you have to shape something that is reported or that will be reported for the first time. Okay, so that is first time. You have to report it for the first time. It should be novel. It should be innovative. Nobody should have done that before. If it is already done, if it is already reported, if it is already published, then nobody is going to take your publication. Nobody is going to publish your manuscript because people will tell there is no novelty. You have just done a repetition of the previous work. So that's why it should be very clear from the beginning that what has not been done. And if something is not being done, that is your research problem. That you can do, that you can report, that you can contribute to science, because there are no reports on this before. Next, it also helps you to define the relationship between your research problem and the body of knowledge in the area. So, what has already been done and reported and published, that is the body of knowledge in the area. So Tulsi's effect on diabetes, Tulsi's effect on cancer, Tulsi's effect on pneumonia, Tulsi's effect on Alzheimer, Tulsi's effect on Parkinson's, Tulsi's effect on different types of diseases are done. But suppose you see that there are no reports on Tulsi's effect on anemia or thalassemia. Okay, so when you are searching medicinal importance, therapeutic importance of Tulsi, you have got so many different types of therapeutic importance. Understood? But you find out that, oh, nobody has reported Tulsi's effect on thalassemia. So then this is the relationship of your research problem with the body of knowledge. So body of knowledge already reports so many things except thalassemia. And that's why you are going to add a new aspect to the body of knowledge through your PhD. What? Effect of Tulsi on thalassemia. Because you have seen already people have reported on pneumonia. Then why will you again do that? So it is refining your problem, defining your problem, and redefining your problem. So initially you thought, I will search the therapeutic aspect of Tulsi. But from therapeutic aspect, which aspect? So these diabetes, oxidant, cancer, and everything already reported. So I have to find out something that has not been reported. So maybe I will report thalassemia. How Tulsi can protect a person from thalassemia. Understood? So like this, you can find out, and this will help you to find out the relationship between your research problem and the body of knowledge. Then improve your methodology. So what is the function of this? Acquaints you with the methodologies that have been used by others to find answers. So as I told you, you want to find out the therapeutic importance of Tulsi. So others have also reported. Some people have reported antibacterial effect of Tulsi. Some people have reported antifungal effect of Tulsi. Some people have reported anti-cancer effect of Tulsi. Some have reported anti-diabetic. So what method have they used for extracting the Tulsi? Understood? The methodologies that have been used by others to find the answers to the question, because they also had the same question. When a person who started his research that whether Tulsi has antibacterial effect, so that person also had the same question: how am I going to extract the Tulsi? So he did some literature search, he already extracted and he reported in a publication. Now you can refer to his paper to find out what methodology he has used for extracting the Tulsi. A person who used Tulsi for anti-cancer, you can refer his paper and find out what methodology he has used to extract the Tulsi for anti-cancer. So like this, for different therapeutic aspect, you know the methodologies. So these methodologies are very important. You can also integrate, you can also incorporate or include in your thesis based on these earlier reported methodologies. Okay, next is tells you if others have used procedures and methods similar to the ones that you are proposing. So you have already thought that I will be using boiling water for Tulsi extraction, and then see if other people have already boiled Tulsi in water. And then if the processes and methods are already used, then see whether this method worked well or there was some pitfalls. Now when you are using boiling water, this can work very well because we know it is called decoction. The method of boiling Tulsi or boiling a medicinal plant and extracting it in hot water, it is called decoction. So decoction is a very well known therapeutic process for medicinal plants, how to extract the medicinal plant. So it can work very well. At the same time, suppose there is a heat labile compound, suppose there is a thermolabile compound. Thermolabile or heat labile means what? That compound in presence of high heat, when you are boiling it at 100 degree, that compound may get denatured. So you might lose that compound because that compound might be thermally unstable. It is not so stable under heat. So when you extract it under heating, under high heat at 100 degrees Celsius, probably will lose all those thermolabile therapeutic compounds. So this is a pitfall. Okay, this is a pitfall. Pitfall means disadvantage, demerit, or where there are chances of getting failed. So these, from these pitfalls you come to know, no, so 100 degrees Celsius heating is not a good methodology. So I should think of heating it under reduced pressure. So then the same thing what you are boiling at 100 degree will probably boil at 60 degree or 70 degree if you reduce the air pressure. So this is how you improve your methodology. Next is broaden your knowledge base in your research area. So you are going to do the research for next few years. Now, when you are collecting more literature, when you are collecting more information, the literature review ensures you read widely around the subject area in which you are intent to conduct your research study. So lots of things you have to refer. Okay, so this is called widely, read widely around the subject area. Suppose you want to develop an idea of Tulsi's effect, then you have to read Tulsi's effect on cancer, anti-diabetic, antioxidant, antibacterial, antifungal, anti-malarial, all. Although your research might be on thalassemia, but in order to get that idea, you have to incorporate all literature in your thesis, in your literature review. And it is important to know what other researchers have found in regard to the same or similar questions. So, the same question that you might be having in your mind, different other researchers might have also the same question in their mind. So, how did they start the research? Where did they fail? And how did they overcome? How did they troubleshoot this? So, this is very important. You understand what other researchers have found out. And then you pinpoint that, okay, they did not get a good antibacterial activity, they did not get a good this activity, probably they used the Osimum sanctum or Tulsi root, and you know Tulsi has more compound in the leaves, so that's why we eat leaves, we eat Tulsi leaves. Okay, so that means there is something that is needed to be done. People have reported from the root, people have reported from the stem, but people have not reported from the leaf. So if I do my research with the Tulsi leaf, then I can inform, or I can actually give an input or contribute to something new to the field. So this is the gap. What is the gap? That extracting the Tulsi leaf for thalassemia. Okay, so this is the gap. So nobody has done this particular thing. Next is contextualize your finding. What is contextualize your finding? The literature review helps identify how your findings compare with the existing body of knowledge. Okay, so you have to discuss, you have to compare. The literature review helps identify how your findings compare with the existing body of knowledge. So already people have reported their studies, already people have reported their data that they have collected because of their research, and that data is available online. So now you have to find out, you have to identify your findings, how your findings compare with existing body of knowledge. So somebody has found that there is 75% inhibition of the bacteria with the help of Tulsi extract. And then you have done your extraction and you have got 90% inhibition of that particular pathogen. So when you compare the 75 with 90, then you find out that your research is far more superior to the existing research, which has reported 75, and you are reporting 90%. So that means the review of literature helps identify how your findings compare to the existing body of knowledge. Okay, so compared to the existing body of knowledge, how your finding is different. How do answers to your questions compare with what others have done? So when you answer your question that I have extracted like this, and I am getting 90%, but he is getting 75%. So what might be his extraction process? So then you find out that there is the gap. You have extracted in a different manner, although the solvent is same, but you have extracted in a different manner. And that person has also extracted in a different manner. So that's why your result might not be similar, your result might be varying. What contribution have you been able to make to the existing body of knowledge? So already people are reporting so many things on the plants, medicinal plant. So what new contribution you can give? So you can tell, like people have reported about diabetic, people have reported about cancer, people have reported about different other things like antibacterial, but they have not mentioned about thalassemia. So this is my contribution to the existing body of knowledge. So further, if a person is going to develop a drug for thalassemia, they have to refer your paper. There you have told like which compound in the tulsi extract is probably important for removing or curing thalassemia. And next, the last point is how is your finding different from others? So other people might have used different solvent, you might have used different solvent. Other people might have used different time points, you might have used different time points. Other people must have used different bacteria against which the activity was checked, and you have probably used a different bacteria that is not similar to the other studies. So you have to contextualize. That means you have to discuss your work with others. So when you discuss your work with others, then you find out what is happening over there. Next is procedures for reviewing the literature. So now we have seen what are the ideal characteristic of review of literature, how this review of literature is helping. Now we are going to see what are the steps to do this review of literature. So first step to do the review of literature is search for existing literature in your area of study. Whatever is your area, whatever is your domain, if you're bioinformatics, if you're biologist, if you're microbiologist, if you're biotechnologist, if you're organic chemist. So first of all, you have to search for existing literature in your area of study. So once you find out the area of your study, the title of your thesis or the core objective of your thesis, then you can actually find out more literature in that area. So search for existing literature in your area of study. Next is once you have searched, next you have to review the literature selected. So you have to go through it and each and every literature and find out what is most important in this. Next, very important part of review of literature is development of or develop a theoretical framework. You have to start developing a theoretical framework, and then you start developing a conceptual framework. And finally, once your theoretical and conceptual frameworks are done, then you have to write. Then you will write. Then you are. Then you will write a literature review, and we call it as a review article. So review article and review of literature are different things. When you write all of your collected information in an article form, then we call it as a review article. In review article, you do not do any research, but you collect information from other researchers and then you prepare your article, and that is called a review article. So review article and reviewing the literature, they are not same. So let us see one by one. Now you want to develop a medicine from natural product against cancer. You want to develop a medicine against cancer. Just a minute. Yeah. So you have to develop against cancer. Now, when you start, you should have at least some idea, broad subject area. What is my broad subject area over here? Anti-cancer activity of curcuma longa. This is my broad subject area. So there are different types of cancer. Now you are not specifying which kind of cancer you are curing. There are different types of different species of curcuma. At the same time, curcuma longa has different parts like stem, leaf, root, flower. You are not defining which part you are going to take. So, some idea, you are not giving whole idea, but some idea you are giving. What is your idea? Anti-cancer activity of curcuma longa. This is your area. Now, start with at least some idea of the broad subject and of the problem you wish to investigate in order to set parameters for your search. So now, once you have got this, you understood that anti-cancer activity of curcuma longa I want to do, and then you set your parameters. Like if I want to search this anti-cancer activity of curcuma longa, am I going to put it wholly, the whole sentence, or I have to pick up the keywords and then search? So all these things are very important to set the parameters for your research. Now, see, whenever you are doing literature search, you do not or we do not do search on Google. This is what? This is called Google Scholar. This is particularly called Google Scholar. So, scholar.google.co.in. Now, in this, as I told you, initially you should have a broad idea, some idea. So suppose you are a student of chemistry, you are a student of microbiology. So none of us are taught about the cancer. So first of all, we should know about cancer. What is cancer? What is cancer doing? What are the different events that are happening during cancer? So that's why you give a keyword, the hallmarks of cancer. Okay, you start giving a keyword, hallmarks of cancer, the keyword over here. So Google will pick up this keyword, that is hallmarks of cancer, and give you all papers that are related to hallmarks of cancer. All papers. Now, here you get one paper, here you get another paper, second, and here you get third paper. Now, when you find out these, first and foremost thing is, do I select all three papers, or I need to select only few papers, one or two papers among these three? Now, when you are selecting this paper, you have to look into different metrics of this journal, of this paper. Now, if you are looking at these metrics, the hallmarks of cancer, this has got citation. Citation how much? 23,804 citation, you can imagine. 23,804 citations to hallmark of cancer. What is citation? Citation means when you are doing a research and you are publishing, then people from around the globe will try to refer your paper. And after referring your paper, they will cite that in their research and then tell we have got this idea from this, or they will compare your result with their result. So it is very important that when you are doing your research, then you see how many times it is cited. Okay. So the more the citation, that means more it has been used for different other groups. Now here you see the hallmark of cancer. This is the first one, and it is cited by 23,804. So that means from this one research, 23,804 people have got new ideas and they reported it, and they have started new research based on this. So whenever they will start a new research based on this, they will cite this article. And how many times it is cited? It is cited to 23,804 times. So that means 23,804 times this research paper is referred by other people. Next, if you look into this paper, Hallmarks of Cancer, the Next Generation, here you can see it has been cited by 18,195 times. Now the question is, which is superior? The more citation is superior or something else? Now, by considering it at as it is not good because it is not cited more times, you will lose a very valuable paper. Why the citation is more over here? Why the citation is more over here and less over here? What makes this possible? How is the citation less in this paper, in the lower paper, second paper, and why citation is more in the first paper? The main reason can be the year of publication. Now see, it has been 2000, means they published it in 2000. So in last 26 years, so now it is 2026. In last 26 years, it got 23,804 citation. But if you see this was published in 2011. So that means it got only 15 years. It got only 15 years to be referred by others. So do not by mistake remove any good paper from your literature search. It is very good paper. But since it got only 15 years and it got 26 years, so 15 years were cited less. Citation means people will discover that paper, explore that paper, and then they will use that in their article, and then at the end they will cite it. They will give credit to the authors, original authors. Next, you can see Hallmark of Cancer. It was published in 2010, and what is its citation? It is cited by 575. 575. Now, whether you are going to take this paper or not. Now see this in 2011 and now it is 2026. So 15 years was there. Now over here, this paper was published in 2010 before this paper. And now there would be hundreds of papers like this, hundreds of papers following them. But nobody has cited it. So that means that is why its citation is 575 because nobody has cited this. So it is the citation number has gone down. It is 575. Next, you know that curcumin, a particular like turmeric is used in our daily food. Whether we use dal or some any other food item, food material, turmeric is almost an integral part of the Indian food system. So you want to see whether these natural products like vegetables, fruits, and other things, they can indeed protect us from cancer or not. So you search vegetables, fruits, and cancer prevention, a review. So then you will get this review, and after getting this review, you find out what has been done or what component in that extract in those uh, vegetables, fruits, and other extracts are able to prevent cancer. So this will give you an idea. Next is another keyword is turmeric and curcumin, biological actions and medicinal importance. So since you want to write on turmeric, you have to find out which compound exactly within turmeric is able to inhibit so many different types of pathogens. Okay, or so able to inhibit so many different types of cancers, so able to inhibit so many different types of fungal diseases. So this you have to find out based on the turmeric and curcumin's nature. So what you have done, you have given a keyword turmeric and curcumin, biological actions and medicinal applications. So people have already reported their biological actions and their medicinal applications. So you don't have to repeat it again. Already people have reported. And then you take this and you can cite it. So these papers which are cited multiple times, they are very good papers and you can refer them. Next, how to compile a bibliography. So once you have collected all this literature, the literature based on the books, journals, publications, articles, reviews. so many things. So you compile them, you club them together. Now you can refer to books also, you can refer to journals also. But when you are referring to books, generally you get the idea about different other aspects, but in journal when you are searching you get pinpointed areas, because each and every researchers they publish their findings in the journals, but book is summary of everything. It is big, but journals are like small precise articles where the exact research that you want is already published. So be aware that sometimes a title does not provide enough information. Then how will you know that which article is good and which article is not good? Because title does not provide enough information to decide if a book journal is going to be used or not. So by looking at the cover, by looking at the title, you cannot decide it. Start with the latest issue. So whatever latest issue you have of that journal, then you start with the latest issue, examine its content, page to this page to see, examine its contents page to see if there is an article of relevance to your research topic. So when you have the latest issue, then you see the content, the outline. If that idea matches with your idea, your research topic, then definitely you can carry forward with it. And then start by reading the abstract. So once you read the abstract, then you get some idea. Understood? So once you get that article, then you have to read the abstract. And if it is relevant, then download and read it. You don't have to download and read it, you just read it online. And if you see it is relevant, then only you open your means once it is relevant, this particular literature is relevant, then only you open that PDF and then you start reading through that abstract, and then you download and read it for the rest of the manuscript, rest of the part. Now here the citations. So first criteria over here is citations. So as you have seen that you select the articles to be reviewed. When you are reviewing the article, how will you select, how will you know that this article is good or this article is not good? Based on citation. First criteria is citation. Now here you can see there is one particular article, and here you can see there is another particular article. Now both these articles they have the same name. See the problem. Both the articles they have the same name. But why or how will you select one particular article from this? Now once you have two articles which are even having same name, okay, same title, but still what you can do is you have to search, you have to find it out with the help of citation. Okay, so citation. So here you see there are no citations at all, but here you can see there are 27 citations. So that means this article is good. People are referring to this article. So that's why they have cited 27 times. But this article is not so good. So that's why people are not referring. It has got no citations till now. Next is impact of natural product on developing new anti-cancer agents. So this is again our another keyword that we have given to find out more information about curcumin and cancer. So whether the natural product, whether the compounds isolated from the plants, medicinal plants, they can cure cancer or not. So you get a paper, you get an article from Track, and you see that it has been cited 698 times. So when it is cited 698 times, you come to know by closing your eyes, you can be very sure that this article is a very high quality article. So people have cited it 698 times. So this is one very important criteria, citations. So those articles which have no citations, they are not so good. But again, you have to think about the date when the article was published. So if an article that was published 11 years ago and it has not got citation, and if an article that was published just 1 year ago and it has got one or two citation, that 1 year ago publication is much better than the 11 years previous article. Now, once you have selected this article, vegetables, fruits, and cancer prevention review, and you have got it citation wise, also this is very good. So you have selected an article. The name is also good, vegetable, fruit, and cancer prevention review. So everything it has clubbed up. At the same time, it has been cited by so many people, cited by 2,446 people. So you feel like this article is very good. Then what will you do? Then you open the article, and after opening, then you go through the abstract. Once you feel that the abstract is important and it can be of use in your thesis, then you can use that article. Otherwise, don't go for forcibly taking the article. Otherwise, if you go on downloading 200 articles, it will be too much. 300 articles, 400 articles, it is too much. So that's why you download only those articles which are sufficing two characteristic. One is citation, so they have lots of citation. At the same time, they have got a good abstract also. So this is very, very important thing. Now the next criteria is impact factor. Impact factor. So what is impact factor? It is number one ranking value for for scientific journals. Okay, so each and every scientific journal will have an impact factor. Now, who gave this impact factor? Who reported this impact factor? In late 1950s, Dr. Eugene Garfield he introduced the terminology that is impact factor. And now the Thomson Reuters had taken up this impact factor. So Thomson Reuters used every nearly in the month of June or July, they used to publish the impact factor of different journals. And then you have now the Clarivate. Now even Thomson Reuters they don't give. Clarivate Foundation they gives the impact factor of each journal. Each journal will get impact factor. Now impact factor is a benchmark. Impact factor is benchmark of a journal's reputation. If the journal has no impact factor, that means there is no reputation. If the journal is having more impact factor, that means there is reputation, high reputation. So it reflects how frequently peer-reviewed journals are cited by other researchers in a particular way. The impact factor helps to evaluate a journal's relative importance, especially when compared with others in the same field. So you find out whether your journal, whether your article is good or not. And next, when you have the article in your hand and you want to submit to a good journal, how do you know whether a journal is good or not? Based on the impact factor. The more the impact factor, the more better is the quality of uh, papers, the quality of the journal, the quality of the reports, the results are much better. Similarly, if you consider the impact factor, it is basically the average number of citations counted in an impact factor year Y for articles published in the previous 2 years. So suppose I want to find out the impact factor today, what I have to do, I have to find out impact factor basically the average number of citations counted by the impact factor year Y. So whenever you want to find out the impact factor, you have to find out what is the total number of citations. Okay, average number of citations. Suppose I have four, the impact factor is basically the average number of citations counted in the impact factor year Y. So in 2025 or 2026, how many citations that journal has got divided by how many articles were published in 2024 and 2023. So if you want to find out the impact factor, that is the number of citations it has received that year divided by total number of citations or total number of articles that were published in previous 2 years. And citations of the articles are loosely defined. In general, they include original research articles and review articles. articles. So they can be both. They may also include editorials. Editorials means editorial will give some information. Editorial will give certain ideas. So they will include that editorial and lengthy reference list. So lots of references. Now, what are the JCR or Journal Citation Reports in cities like in cities journal citation report or this journal citation report or JCR, this is called JCR. JCR is given by Clarivet, and every year, every year the journal, every year the means JCR or the Clarivet they gives the particular metrics to the journal. So we have site score and we have site score rank, and the site score is calculated, and then the average number of citations is received by the calendar year by all items published in that journal in the preceding 3 years. This is the formula of site score. Similarly, you have site score rank also. At the same time, you have another that is called source normalized impact per paper. This is called SNIP, source normalized impact per paper. A sophisticated metric that intrinsically accounts for field specific differences in citations. So you might think, oh, the biology people, they have got lots of citations. They generally get their journals have lot of citations. Some people will say we are from statistics and we do not have good citations. So are our journal bad? No, your journal is not bad. Now you need to see how many articles are accepted by the journal, how many articles are published, and based on that, you have to find out the citation and the impact factor of this. So based on the area, based on the journal, the impact factor will again vary. So it all depends upon the field. So that's why nowadays people get this impact factor based on SNIP, source normalized impact per paper. Next is Scimago journal rank. This is also a very good example. So if a journal is already indexed in Scimago journal rank SJR, then you can publish in that. No problem with that. So Scimago journal and country rank is a portal. So there is a rank is portal that is called the Scimago journal and country rank. It is a portal that includes the journal and country specific indicators developed from the innovation information contained in the Scopus. So whatever indications, whatever informations are there, so those informations are collected, and based on that, the journal is given a rank. So that is called Scimago journal rank. So each and every journal from Clarivet, they get this impact factor. Now, if you consider in 2025, you can see there are so many different types of journals, and different journals have got different impact factor. So if you consider these impact factors like American Cancer Society by Wiley. So they have a journal called a Cancer Journal for Clinicians, CA. Now see this impact factor, 232 impact factor. Okay, there is another journal from Elsevier called Lancet. Its impact factor is 104. Now, on what basis you can see the impact factor varies? Now, this journal started publishing their paper in 1950, and this journal started publishing their paper in 1823. So, which one is old? The Lancet is very old. Lancet is very old. So, ideally, Lancet should have got much more citations and Lancet should have got much more impact factor. But compared to Lancet, this journal, which started in 1950, this is in 1823, this is 1950. This journal started much later, and this journal has got much more impact factor. Why? Because it is open access. This is also a very important criteria for getting the impact factor. So this is an open access journal. Why open access is important? Because people can freely refer to the articles. Each and every person can freely refer to each and every article. So they don't have to get subscription, they don't have to pay for that. But this article or this journal is publishing articles, and this journal is published by Elsevier, but this is not an open access journal. You should have subscription. If you do not have subscription, then you have to pay. Like this, chemical reviews 55.8, chemical society reviews 39.3, nature 48.5, science 45.8. So all these journals, they are actually very important because they are very high impact factor, and all this impact factor is based on how many citations they are getting. Based on the citations, the impact factor will vary. Now when you are looking at a journal, then suppose you have got an invitation and you feel like, oh, this journal is very good, American Journal of Pharmacological Sciences. The name is American Journal of Pharmacological Sciences. So that means it is an international journal. So we can publish our paper in this. Whatever research article or review article you have created, you have written, you have drafted, you want to submit to this because this looks as a very good journal. At the same time, the website is also very nice. It is a good looking website and everything. But see, what do you think? Can you submit your article? Do you think this journal is very good to submit your article in this? First of all, there is no impact factor in the homepage of the journal. This journal, although it is an American Journal of Pharmacological Sciences, they are referring to the name. We do not know whether it is truly American or not, but at least this journal does not have any impact factor on its homepage. All journals should display their impact factor in the homepage, but there is no impact factor over here. Similarly, see, there is no editor-in-chief of this journal. It is written editor-in-chief, apply for this position. So editor-in-chief is not there in the journal. So they're asking if you want to become an editor-in-chief, you can apply. So this cannot be a good, authentic journal. Now, if you have this particular journal, like chemical reviews, and its impact factor, then if you see in 2020, the impact factor, this is the American Chemical Society's publication. So there are publishers. So this publisher is American Chemical Society. Now, American Chemical Society has published a journal, and the name of the journal is Chemical Reviews. So in the homepage only, it is displayed that the impact factor is 60 point. This impact factor in 2020 it is 60.622. So 60.622 is the impact factor of that journal, and it is displayed in the homepage. So there is no question of doubting it. You can tell, okay, this is very nice and this is a very good, authentic journal. What are the other criteria whenever you are selecting a journal for downloading your articles? Next thing is, it should be indexed in these databases like PubMed and PubMed Central. So PubMed and PubMed Central, these are the databases, and here the journal should be indexed. If it is indexed in this PubMed or PubMed Central, then that means it is a very good journal. It can be indexed in Medline, American Chemical Society's Chemical Abstract Services, that is called CAS. Then Science Citation Index Expanded, that is SciSearch, and there are different other bodies. And most important is Scopus. Okay, most important is Scopus and DOAJ. So there are different types of databases. These journals should be indexed in that. At the same time, the journal should subscribe, or journal should be following which kind of principle? The Committee on Publication Ethics, that is COPE. Who will become the author? Who will become the co-author? who cannot become the author. So all these guidelines are given in Committee on Publication Ethics, that is COPE. So once you select this journal based on the indexing, based on the impact factor, based on the citation, you can download the article. Now see, suppose this is the title, Anti-Cancer Potential of Plants and Natural Products are Reviewed. This is the title of your search. And then once you want to confirm that it is present in PubMed or not, so you open the PubMed homepage, and on this PubMed homepage, you just copy paste the name of the article, and then you see whether the article as well as the journal is indexed in PubMed. So when you add this, you can see there is a different article that is in PubMed. There is another different article that is in PubMed, and there is another that is in PubMed. So you get all these articles that are already indexed in PubMed. So if your article is not coming in PubMed and it is a medical research, then that means there is some problem. So PubMed should be there. Now, what we found one article from which we wanted to download and write our review. So impact of natural product on developing new anti-cancer agent. So you got this article. It was in Chemical Reviews 2009 July 109. So this has been published. This article is published. It has got a PMID, PubMed ID also. And now it is coming in PubMed. As I told you, it is a Chemical Reviews. Chemical Reviews are very high impact factor, and it is under the very well-known publisher. So that's why it is indexed in PubMed. Now, once you have selected the literature based on what? Selected the literature based on the citation, selected the literature based on the journal's impact factor, and all these things. Then what you have to do? Then you have to review the literature that is selected. Suppose you have collected 50 papers. So then what you have to do? You have to review, you have to go through each and every paper. Okay. So now that you have identified several books and articles, the next step is to start reading, and then reading them critically to pull together themes. So in those 50 papers, suppose they have worked on Tulsi. So you pull together what kind of extracts were used, from where Tulsi was extracted, whether it was from America, or it is from the Europe, or it is from India, or it is from other Asian countries. So from where Tulsi was collected, from which parts of Tulsi were taken, and which solvents were used for preparing Tulsi extract. So all these things you have to read, then you make the themes, you get new ideas, and then you start finding out everything. If you do not have a theoretical framework or themes in mind to start with, use a separate sheet for paper for each article or book. So next thing is theoretical framework, the sections. So how will you do the sections? So suppose by reading, when you are reading these articles, then you get new ideas, and then you can make your own sections. If you do not have, then while reading the articles, you take a pen and paper and write down what sections can be done. Like in solvent extraction. So in solvent or in extraction, you can add 7-8 different types of extraction. In case of antimicrobial or anti-cancer, you can do some different activities. So you can put it over there. So this makes your theoretical framework. Next, you slot the findings from the material. Whatever material you have got, so you sort it out. And then you give them, you include them under different themes, under different headings. And where are the differences? Where there are significant differences of opinion among the researchers, give your opinion about the validity of these differences. So suppose there are two different opinions between the researchers, then whatever you think, you can put it over there. Then ascertain the areas in which little or nothing is known, the gaps. So whenever you are having your own review article, whenever you are writing your own review article, you should ascertain the areas in which little or nothing is done. So you are doing this, so you are contributing to this. So give your own area. Now see what was our topic or target. So we took one case, and according to the case we are moving towards. So what is the first case? What is the first thing that we saw or we wanted to do? First thing is turmeric and cancer. That is your research. Your research is on turmeric and cancer. Now different people have reported, different people have published different paper. So there is one important paper maybe, that is how turmeric can cure brain tumor, how turmeric can cure breast cancer, how turmeric can cure gynecological cancer, how turmeric can cure thoracic or HN, how turmeric can cure melanoma. So different people, different research groups, they have reported different cancers. And which compound of the turmeric? Curcumin, one single molecule. See, one single molecule can prevent, can stop so many different types of cancer. So then you prepare this chart. This chart from whom? This chart from by referring different papers. So this is called theoretical framework. That means your review article will have so many different types of cancer, and then how curcumin is playing an important role in inhibiting this cancer. So this will become your theoretical framework. So what should come in the theoretical framework? First and foremost is a title. A very good title should come in your theoretical framework. What is the second thing? The abstract. The abstract is very important. So you should have a very good abstract after title. Then you have an abstract. Then you have an introduction. You have an introduction, and that introduction will give the briefing that what you are going to do, why you are going to do. So in introduction, it is very important. In one paragraph, it will tell what things you will be doing and why you will be doing this. And then there is composition of turmeric. So in turmeric, when you extract it, what is present in turmeric? And there you find out curcumin is most important component of turmeric. So what you have to do, you have to find out the curcumin analogs. Are all plants having the same one molecule of curcumin, or there are analogs? And then you will see that curcumin has number of analogs. And then you just draw their structure and write down what are the different types of curcumin. And then find out what are the uses of curcumin, where curcumin is used, how curcumin is used, all these things. And finally, you conclude that since curcumin has so many effects, so much effect against cancer, and curcumin is non-toxic, so curcumin can be used for preventing cancer. So different types of cancers, curcumin can be used. So this is very important on point of view. So you can then conclude like that. Now after this, you have a conceptual framework. So what is conceptual framework? When you are developing your theoretical framework, when your theoretical framework is done and you are referring to the papers, you get new ideas. You get new ideas click in your mind. And then based on those new ideas, you can develop your own framework, and that is called conceptual framework, a new concept. People will get new idea from your work. So each and every paper, suppose a paper is reporting how curcumin can protect, how curcumin can inhibit bone cancer. So they will tell that curcumin will go and bind with this molecule of the cancer cell in case of bone cancer. So they will report some molecular target. Some genes will be upregulated to prevent cancer. Some genes will be downregulated. Similarly, in case of oral cancer, a paper that has reported oral cancer, that paper will give a new molecular target, that curcumin goes and bind with this, and then curcumin inhibits these genes, curcumin upregulates these genes, and this is how curcumin is protecting against oral cancer. Similarly, curcumin is reported for brain cancer or brain tumor, so it will give some new idea that curcumin is going and doing what. Then you know that curcumin, in order to act with the cell, curcumin will go and bind to the receptor first. So what are the different types of curcumin receptors? And then you can, from all those 200, 300 papers, you can make one paragraph on curcumin receptors. And then disease targets of curcumin. So which diseases curcumin can target? And what is the mechanism of action? What exactly curcumin is doing? What is the true mechanism of action? What is curcumin's work over there? So then you make your conceptual framework. This is an example of beautiful example of conceptual framework. What is it doing? You can see curcumin is in the center. So from where you have got curcumin? Curcumin you have got from the turmeric extract, from the turmeric. So the turmeric plant has its integral component called curcumin. And this curcumin, this one molecule of curcumin can modulate, I am referring to the word modulate, or impact so many different types of genes in a human body. Curcumin, just a single molecule of curcumin, can go and control so many different types of genes, and all these genes are related to cancer. So these brown genes, they are cancer causing genes. The brown genes, they are cancer causing genes. So when these genes are upregulated, then cancer is caused. So that's why what curcumin is doing, curcumin is down regulating these brown genes. Molecular targets down regulated by curcumin. So all these brown genes, which are very highly expressed in case of cancer, as soon as curcumin goes inside, curcumin will start down regulating all these brown genes. Just one compound, that is curcumin. At the same time, these genes like P53, NRF2, PPR gamma, they are very significant to stop cancer. So during cancer, they are down regulated. The cell down regulates so that cancer can be caused. And when curcumin comes inside, then they upregulate these blue genes. The blue genes will be upregulated. Upregulated means more synthesis of the blue genes, more expression of the blue genes. So if there is more synthesis or more expression of the blue genes, and these are anti-cancer genes, so that would be good. It can prevent the cancer. It can stop the cancer. So this gives you the conceptual framework. So the conceptual framework is not by one particular paper, but all these 200 papers. From all these 200 papers, you have got this concept. What kind of concept? That what are the molecular targets of curcumin. So just by reading your review article, just by seeing this picture, a person will get all kinds of information that have been done or that have been reported in last means few decades. In last 30, 40 years, whatever people have done on curcumin, just by looking at this image, just by looking at your theoretical and conceptual framework, a person will get whole idea. He or she does not have to read all those 200 articles, but by looking at your figures or reading your article, that person will come to know what research has been done in last 200 years in the area of curcumin and cancer. So what is a review article? Suppose you are cooking. Okay, your mother has put rice and she tells, can you go and check whether the rice is cooked or not? So what will you do? You will go and you pick up a grain from the rice bowl, press it and check, this is cooked. Then you pick up another grain, press and check, this is cooked. You pick up another, this is cooked. So are you going to do this thing for all thousands of grains of rice that is boiling in the container? You're not going to do that. What will you do? You will just pick up one grain, you will just pick up one grain, press it, and you know what is the condition of the rice in all, what is the condition of all rice in the container. You pick up one grain, just you pick up one single grain of rice, press it, you come to know what is the condition of all grains in the container. So, review article is like that. People will just pick up your review, one article, and they will get whole idea about 200 researches that have already been done in that area. They don't have to go through all those 200 research article one by one. But just by going through your review article, by one review article, they will get the entire idea about all those 200 researchers. So that is why review article is very critical. Review article is very important and very, very significant for all researchers. So suppose you read 20 review article. Reading 20 review article is equivalent to reading 4,000 research papers. Okay, 20 review article is equivalent to reading 4,000 research papers, because one review article will cite 200 article. Like this, you have read 20 article. So 20 into 200, 20 multiplied by 200, around 4,000 research paper. It is how will you complete 4,000 research paper if you go on reading them? You cannot. But just by reading those 20 articles, you can cover 4,000 research articles. So this is the added advantage of reading review. Review gives you idea, and reading one review article means reading almost 200 research articles. So this is now the conceptual framework and mechanism of action. So from different articles, you have got that curcumin binds with GPCR, then it gives stimulus or it triggers IP3R, and then IP3 gives the trigger to the ER, and then particularly this gene is expressed or downregulated. Either it is upregulated, downregulated. Growth factors, they give signals to JNK chain, and then after that this gives signal to AP1, and then AP1 gives signal to the other receptor, other molecules, signal molecules. Similarly, TNF, tumor necrosis factor, binds with the tumor necrosis factor receptor, and finally it activates IKK, and then IKK mediated this thing. So this means that whenever curcumin is applied, whenever curcumin is taken in, then it is modulating the gene expression, and because of the gene expression, there is the receptor ligand binding, and this particular receptor ligand binding is resulting in further gene expression. So this is how the mechanism of curcumin mediated anti-cancer activity is happening. So the conceptual frameworks give you this particular mechanism of action. Once you have your theoretical framework and conceptual framework, then you start writing the literature review. Start writing your review article. So you can see there is a title over here. After the title, you can see the name of the authors. After the name of the authors, you see the subsections. So these subsections are the theoretical framework. And under these sections and subsections, there are more sections. And then you have this progression. So it will make a story. It should be like a story, a free flow. Like from this will introduce you to this will introduce you to this will introduce you to this. So it will be a story. Then how to write the references? Then there are different citation styles. So among these, APA, MLA, and Chicago, these are the mainly different styles of referencing. So whenever those literature that you have collected, you have to write it. So if you have a book, you can write like this. Ebook, you can write like this. If you have an article, you can see this is the surname, this is the name, and then this is the year, then the name of the title of the paper, then the journal, then this is volume, and then this is page number. You can also have this volume and issue number. After 14, there is an issue number I, and then you have the page number. So these are the different types of reference style. But mostly the American Psychological Association, that is the APA style, is referred. You can see there is the surname, then the name, abbreviations, then you have the year, then you have the title of the paper, journal, volume, and page number. APA can be modified by different journals. This JOC, now see, they are putting the APA, but something is missing over here. What is missing? The title of the article is missing. And they have abbreviated the title of the journal. The name of the journal, they have abbreviated, title of the article is missing. Similarly, over here, you can see the title is there. But after 1, 2, 3, 4, 5, they are putting et al. If there are more authors, they are putting et al. Here there is no issue number, but here there is putting issue number. Here they have put comma, here they have put colon or semicolon, and then, sorry, they have put colon, and then they have put the page numbers. So every journal, they have their own style, and they can modify the APA style. So if you see this, you can have this particular reference. So beautiful title. See the title of this review article: Curcumin and Cancer, an old age disease with an age old solution. Very nice title. And see how many articles they have referred: 282 articles. So if you read this review, it is equivalent to reading 282 articles. You don't have to go through each and every article, 282 article, but if you read this review, and it will be done. This is the name of the journal, this is the volume number, this is the year, and this is the page number of this. So with this, we'll conclude. So as you have seen, that review article, writing a review is a continuous process. So you have A, B, C, D, E, F. So in your chat box, can you tell me, can you arrange it properly? And you can tell the sequence, give comma, like first maybe it is A, then comma B, then comma C. So according to this, you just arrange and let me know whether you have understood the process of review writing or not. Okay, so review article is a continuous process, like making this Ganpati. Okay, so if you start Ganpati, you have to collect the mud, and then you make the framework. Then you add up one by one different parts of Ganpati, and then finally you get the whole Ganpati. So if you have understood this process, the flow chart of Ganpati making, it is like similar to making a review article. So please write down or arrange this. If you have any questions, then please ask me so that I can answer you.
Thank you, sir. Thank you very much for such as fruitful sessions from your sides.
Thank you. Thank you, sir.
Thank you, sir.
I think there are no comments in the chat box.
Yes, sir. I think student haven't any query from your
They
side.
can, yeah, if they have any query, then they can reach me anytime. Uh, so my email ID is there. They can reach me. So if they have any
Sure, sir. Okay. Thank you, sir.
query. Okay. Then we can conclude, right?
Yes, sir. It's a
Okay. Thank you.